So here we're beginning chapter 18 from our evolution textbook, and this chapter is titled Evolutionary Medicine. Seems like a good sort of capstone chapter to end our class with. Now what we'll see in this chapter is not only are we challenged medicine-wise from the ways in which viruses and bacteria evolve, and of course they present particular challenges to how we can cure diseases caused by those microbes. But also there's medical issues that arise from our own evolutionary history, who and what we are based on our genetic heritage. And we are vulnerable to a variety of ailments because, in a sense, we are maladapted. We are not well adapted in some respects, and that leaves us vulnerable. So we'll address that as well. Now, the opening story here in our chapter talks about the Lassa virus in West Africa, and the fact that it's one of the most deadly viruses known to humans. Only about 50% of those who contract the Lassa virus actually survive. Interestingly, about 34% of people in Nigeria, West Africa, carry an allele at the large gene, as it's called, whereas outside of Africa, almost no one carries that allele. Right? Head scratcher. There must be some evolutionary reason for that, or at least we might suspect so. One of the things that we have learned is that this allele originated about 1,000 years ago, so relatively recent. Now, it's also recently been discovered due to various outbreaks from this virus that it is transmitted not from human to human so much, but specifically from the African soft-furred rat to humans. And as your introduction talks about, there's a couple different mechanisms by which that might happen. So that's the overall story, a very interesting one, which very often we return to these introductory stories later on in the chapter. But as an overall general kind of message going into the chapter, pathogens, and I sort of mentioned this at the beginning of the slide, often undergo rapid evolution. And that makes it, of course, particularly difficult for host immune systems to keep up. So we have, once again, what's often referred to as an evolutionary arms race between pathogens and us, with the pathogens really having the upper hand because of their rapid evolution. And also, as I mentioned above, humans are often vulnerable to certain diseases, making us maladapted as opposed to adapted. So let's dive in here and learn some things and cover some things about evolutionary medicine. Now, of course, you've heard about natural selection ever since you took Bio 181 or earlier, and certainly natural selection has popped up over and over again in our evolution textbook from different angles. Interestingly, though, in this topic of evolutionary medicine, we're discussing the limits of natural selection. And of course, this section is titled Medicine and the Limits of Natural Selection. There's numerous aspects of our human anatomy and or physiology that are, quote, maladapted. They certainly are not an optimum sort of phenotype given the costs of what they represent. So your textbook uses the example of the human eye and how it represents several examples of maladaptation. Hereditary nearsightedness is one of them. A large proportion of people over the age of 50 especially suffer from nearsightedness, and it does have a genetic basis to it. Of course, I'm one of those. Cataracts, which involves damage to the lens proteins of our eyes. I think by the time you're reaching your 70s or so, a very large proportion of people have cataracts. And then there's the classic blind spot in our eye where the optic nerve connects to the retina. And our eye, we don't even notice it, has to sort of dart back and forth in order to make up for that vision impairment. So there's a classic example of maladaptation. I went on the internet, found a visual to sort of represent nearsightedness and farsightedness, etc., and how that occurs. So you can check that out in the bottom corner if you want. But the other aspect here is that for various maladaptations, there are 6 evolutionary explanations, possibilities, alternative hypotheses for why a maladaptation might occur. So one of the big ones is that pathogens evolve faster than hosts do. And given our long generation time as human beings and relatively low numbers of offspring per reproductive effort, pathogens like bacteria and viruses completely blow us out of the water and, of course, evolve magnitudes faster than we do, as an example. Another aspect, number 2 listed here, is that natural selection can lag behind evolutionary change. So, of course, it takes an environmental change. It takes an environmental change to happen first. Then, if there is genetic variation that might be favorable, natural selection might then happen to change the genetic makeup of a population. But there may not even be a genetic variation that might be matched to an environmental change, in which case natural selection would really lag behind. So that's another major component. There's also evolutionary trade-offs that make adaptation nearly impossible. So, whereas our upright posture as humans carries advantages. The original adaptive reason for bipedal upright posture is still debated, but of course we're able to have better access to our opposable thumbs and grasping hands for things like tool making. But being bipedal also means that we're more limited in our locomotion than something that is quadrupedal. There's lots of examples of these trade-offs. A turtle, for example, a turtle shell is a great adaptation for protecting it from potential predators, but it obviously slows the animal down, or if it's flipped upside down, it's vulnerable to dying. So trade-offs. Then there's historical constraint, which means that we are trapped by our evolutionary history. And even though we might sort of hypothetically dream of a particular adaptation that would be really cool, organisms, including animals, are sort of constrained to develop in specific ways. They're bound to develop genetically in specific ways and can't get out of that rut, if you will. Take whales, for example. So clearly whales would benefit greatly by having something like gills. So when a sperm whale dives down a couple thousand feet in the ocean to go look for squid, it would be wonderful if it didn't have to swim all the way back to the surface to exhale, or they really have exhaled, and get a fresh breath of air. But due to historical constraint and the development of the entire anatomical and physiological systems of whales, it's probably really not possible for whales to be able to adapt in that way. Second to last, some traits increase fitness at the cost of disease vulnerability. So we'll take a look at that as well in this chapter. And then what might actually appear as a disease actually could be an adaptation after all. So we'll take a look at that too. So I think on the last slide, the first of 6 possible explanations for maladaptation had to do with the rapid evolution of pathogens. And so we can take ourselves, of course, as an example of this. While our immune system is marvelously adapted, we essentially have the immune system of most jawed vertebrates. The rapid evolution of pathogens, bacteria, viruses, makes us particularly vulnerable. So, for example, as our immune system works to destroy one viral variant, natural selection may then favor a mutant variant that can evade our immune system and get around it. So that's a limitation. Of course, as you're well aware, viruses have a very high mutation rate because they do not have any mechanisms to repair their DNA or RNA. So those mutations stick and are not corrected. Lots of mutations. And there's the fact that viruses in particular have extremely rapid reproduction. So case in point, in one day, an HIV virus in a human being can give rise, a single virus can give rise to 1 billion descendants. That's pretty daunting. And then the 4th aspect listed here that makes it challenging for our immune systems to cope with the rapid evolution of pathogens is horizontal gene transfer. So viruses can take up genetic material as they replicate, and of course bacteria are well known for horizontal gene transfer and can accumulate large batteries of genes that confer resistance. We'll come back to that in a couple slides. Collections of genes that are referred to as cassettes. So a case in point here that's on the slide with the figures is involves viruses that were sequenced from 9 HIV patients at 4 months, at 5 years, and at 8 years to be able to get a temporal aspect of how the viruses are evolving within the patients. SNPs, single nucleotide polymorphisms, were tracked in one patient and in one particular region of the virus called the P17 region. And if you look at the figure in the top right, it shows you the base pair position in this region between base pair 0 and past 300. And then the frequency of HIV single nucleotide polymorphisms that arose within one patient. So you can see after 4 months, there are 6 SNPs that are plotted there. At 5 years, there are many, many more. And at 8 years, of course, many more than that. So these guys are replicating and mutating and evolving rapidly. There's a phylogeny that's shown on the bottom right, and it's pointing out there that there are different regions of the virus, HIV virus, that evolve at different rates. So in actuality, the V3 region evolved much more rapidly over a 98-month period than the P17 region. So there's that aspect as well. Different regions or parts of these pathogens can evolve at more rapid rates than others. So these are some of the challenges posed by pathogens to our immune systems. So this section, some bacteria turn into pathogens within our bodies, is pointing out that when a particular microbe invades our bodies, it may not, in fact, be a pathogen. It may not, in fact, be a disease organism. But if it establishes itself in our bodies, then that environment within us may be favorable for certain mutations to increase in frequency due to natural selection, turning these creatures into pathogens within our bodies. So, in a sense, like the first bullet point is pointing out here, our bodies can act as a flask for bacteria to reproduce and evolve. And you've seen some examples in our semester here of researchers tracking the evolution of bacteria over tens of thousands of generations in flasks. And it's remarkable what kind of evolution happens in those contained environments. So our bodies are a contained environment, sort of a flask, sort of a petri dish for bacteria to reproduce and evolve. Case in point, Pseudomonas aeruginosa. It's naturally found in numerous different habitats, soil and sediments, etc. And it usually doesn't survive if it enters our body. But on some occasions it does. And in particular, for someone who has cystic fibrosis, it thrives in the lungs of someone with cystic fibrosis. The conditions in the lungs, the environmental conditions in the lungs of someone with cystic fibrosis are particularly favorable to Pseudomonas aeruginosa becoming established and reproducing. And what we know, what we observe, what we have seen is that mutations can transform these bacteria from free-living individuals that are harmless into clusters that stick to the linings of the lungs and turn pathogenic. In fact, numerous cases with individuals with cystic fibrosis, the bacteria evolve proteins that wrest or take iron atoms from their host. So that's one particular aspect of their evolution that enables them to be able to survive and reproduce in our lungs. And different areas of the lungs provide different microhabitats in which these clusters of bacteria can evolve. Different populations, if you will, are evolving in different directions in different parts of the lungs. So take a look at the figure on the right-hand side of the slide here, study it a bit to see what it's representing. You get Pseudomonas aeruginosa from water or soil coming into one's body, and you get local populations adapting to different lung environments. And you end up getting infection, so then it becomes a pathogen. And in different parts of the lung, down the left-hand corner of the figure, it's pointing out that divergent, locally adapted populations coexist in the lung. So due to the different conditions in different parts of the lung, different genetic variants of this virus can be evolving in there. So as you probably know, virulence refers to how harmful a pathogen is. And so here we're going to take a quick look at the evolution of virulence in microbes in particular. So when Pseudomonas aeruginosa does invade someone, they start off with flagella, and they use those flagella to swim deep into the lungs. And as they swim into the lungs, they secrete toxins and cells die. So that's what happens with Pseudomonas aeruginosa. Now, interestingly, evolution within different hosts, different human hosts, consistently leads to Pseudomonas aeruginosa becoming non-motile and losing its flagella and becoming less virulent, less harmful over time. So very interesting outcome. It's beneficial for them to do that in the long run, so natural selection shapes them to become less virulent. Very interesting experiment with Salmonella Typhamurium. And this is a bacterium that causes really bad food poisoning. So experiment was done in which mice were infected with a very mild form of this salmonella. And after the bacteria were given the opportunity to replicate, then they were transferred to healthy mice. And this is called a serial passage, transferring these organisms from one host to another. And there were 10 serial passages that occurred in this experiment. And after 10 serial passages, every mouse died. So take a look at the figure in the lower right. So again, remember, when upon initial infection with the salmonella, that the bacteria were not very virulent. But after these 10 replications or 10 serial passages, they became so virulent that 100% of the mice were dying after 10 serial passages events. So there's two primary factors that affect virulence, or that factors affecting virulence that can evolve. And that's what's going on here with the salmonella. So traits can allow better competition for resources that raise fitness. And so those traits are going to be favored by natural selection. Traits that allow, in this case, Salmonella Typhamurium to compete better for resources. So that's one way that virulence can evolve, going along with that being a better competitor. And the second reason, the second explanation for a factor that can affect the evolution of virulence is a higher likelihood of being passed from host to host can increase virulence. And that is definitely the case with the salmonella experiment. Of course, these salmonella were artificially passed from host to host. So they were not allowed to stay within the host and evolve within the host, but being passed from healthy individual to healthy individual to healthy individual, it was beneficial in that case for the bacteria to increase their virulence. So either traits that allow better competition for resources, giving rise to higher fitness, that can give rise to higher virulence, or if there's a likelihood that the pathogen can be easily passed from host to host, then that also can increase virulence. So I'm sure you guys are well familiar with antibiotic resistance. And you may know that in the mid-1900s, scientists, of course, discovered that some fungi and bacteria produce chemical compounds that can kill off pathogens. And those chemical compounds we call antibiotics. It was a miracle of science, if you will. And there were some, including the Surgeon General of the United States in the 1960s, who proclaimed that disease would be conquered by antibiotics. And in the future, there would be far less disease afflicting human beings because of the invention of antibiotics. But resistance to antibiotics was almost immediately observed. And in the course of time, some bacteria have become resistant to all known antibiotics. Pretty mind-blowing and scary. In comparison, insects, of course, have evolved resistance to pesticides, and plants have evolved resistance to herbicides. And so what we can see by standing back and looking at these three examples, bacteria have evolved resistance to antibiotics, insects to pesticides, plants to herbicides. In all three cases, humans have drastically altered the environment with some kind of deadly chemical. So we have altered the environmental landscape for these creatures. And you know how bacteria, insects, and plants have evolved resistance, wink, wink, because of your understanding of evolution. Any genetic variant, whether it's pre-existing or comes about by mutation during the course of this environmental change, is going to be favored by natural selection and passed on differentially. And the population will adapt. So now, last point here, resistance has evolved in to antibiotics under several different conditions. These are some of the ways that bacteria are able to fend off our antibiotics and survive the gauntlet of them. Number one, cells, bacterial cells, can close off the passage to antibiotics through their cell walls, essentially putting up a blockade so that these chemical compounds cannot get into them. Secondly, these bacteria may have or may evolve enzymes that break down the antibiotic, rendering it harmless. And third, some bacteria can actually push the antibiotic out of the cell before it even causes harm. So we know some of the ways that bacteria end up having antibiotic resistance. So onward we go. So here's a remarkable example of the evolution of resistance with antibiotic resistance within a single host. So first of all, let's reiterate that bacteria are able to rapidly evolve resistance for a couple reasons. They have very large population sizes, large numbers of individuals, and they have very rapid reproduction. So at every binary fission event, there's an opportunity for mutation. So that's going on within any host. Now, there's a patient in Norway several years ago who was diagnosed with tuberculosis, and 3 standard antibiotics were prescribed. After 8 months, only pyrazinamide had an effect on the tuberculosis bacteria. And mutations were identified that established resistance for the other 2 drugs. So in response to this, 4 new antibiotics were administered. And a year later, the bacteria were still going strong. This person still had a very active tuberculosis infection. In fact, a hole had developed in his lung. And the bacteria had evolved resistance to 3 of these 4 new antibiotics within this one person. All right, so then they regrouped and decided to administer 2 new antibiotics, and the bacteria were soon resistant to those as well. Finally, linezolid was found to be effective, and the infection finally cleared up. However, the lung had to be removed. Now, mutations had accumulated in different lineages of bacteria, and this is, of course, very common in bacteria in general. But horizontal gene transfer is what led to the evolution of what we call cassettes, bundles of resistance genes that can then be transferred by horizontal gene transfer, cassettes. And that is in large part what was going on among the bacteria causing tuberculosis in this individual. Now, not that you need to know the details of the figure on this page, of course not, but it's worthwhile to look at the timeline and look at the antibiotics and the mutations to get an idea of the complexity of what was going on and how these bacteria were responding to the different antibiotics as time went on. So here we can see a timeline of different antibiotics that were introduced at different periods of time over the past, almost 80 years. And it's really remarkable to see that as new drugs have been introduced, bacteria have inevitably evolved resistance. And the medical community agrees that if bacteria continue to evolve along the same trajectory, by the year 2050, just 30 odd years away from now, antibiotic resistance may cause a staggering 10 million deaths per year. Certainly catastrophic. So one of the key things we need to do is find ways to reduce antibiotic application to individuals or in general. So we have to cut down our use of antibiotics. This is essential if we're going to slow down this trend. So one of the aspects here is that the vast majority of antibiotics are not in fact given to sick people, but they are given to healthy farm animals. And this is indeed a problem because this provides a reservoir for bacteria around the world to be evolving resistant to these antibiotics when they are confronted with that new environment. Indeed, as farm animals urinate or defecate, those same antibiotics can be introduced into the environment in general, and bacteria in the general environment can then be evolving resistance to antibiotics even outside animal bodies. And then secondly, the Centers for Disease Control estimates that about half of all antibiotics prescribed by doctors on an outpatient basis are completely inappropriate. They shouldn't even have been prescribed in the first place. So, for example, if a patient actually has a viral infection, then as you know, that is going to be ineffective, or antibiotics are going to be ineffective. And yet, in many cases, antibiotics are prescribed even though the patient only has a viral infection. So these are a couple key ways that we can at least reduce the tendency for bacteria to be evolving resistance while we try to find ways to combat bacterial infections in other ways.